Good evening. Good evening. Welcome to the Shoals Marine Laboratory's weekly marine science seminar. I'm Sarah Morris, the John M. Kingsbury Executive Director of the Shoals Marine Lab, and I want to thank everybody for joining us this evening for our seminar, our 12th and final Rock Talk of the 2024 season. Our seminars are affectionately known as Rock Talks because back in the early days of the lab, all of the lectures and guest lectures were done literally on the rocks. We've now moved everybody inside, which is a little bit better in terms of, I know it's just, which is better for everybody to be together. Um, but we still use that term to kind of go back to our early history. Our Rock Talks provide an opportunity for people to hear, to come together and hear about a variety of different topics related to marine science and to the environment. For those of you joining us online or anyone who may be new to our lab community, the Shoals Marine Lab is the oldest and largest marine lab in the country that is focused on undergraduate training, both in coursework and in research. The lab is operated jointly by the University of New Hampshire and Cornell University. In addition to our guests joining us remotely tonight, we have two groups of students currently on Appledore. The Marine Immersion class from the University of New Hampshire and a group of students from the Manchester School District taking marine science and sustainability. We're excited to have everyone both in person and online this evening for this seminar. The format of the seminar tonight is a 45-minute talk followed by 15 minutes of questions and answers. Although based on a conversation that I had with the seminar speaker a few minutes ago, if you have questions during the seminar, we can allow the seminar to go long and you can ask questions during the seminar itself. That's true for anybody on Zoom as well. Put your questions in the Q&A box at the bottom of your Zoom screen. And we will end promptly at 9 p.m. This evening's seminar speaker is Dr. Jennifer Miksis-Olds, who is a research professor at the University of New Hampshire and is also the founding director of the Center for Acoustics Research and Education at UNH. She completed her bachelor's degree in biology at Harvard University, her master's degree in biology at the University of Massachusetts Dartmouth, and her PhD at the University of Rhode Island in oceanography. She also completed postdoctoral work in bioacoustics and oceanography at the University of Massachusetts Dartmouth. Dr. Miksis-Olds is a member of the scientific committee of the International Quiet Ocean Experiment Program and serves as a scientific advisor to the Sound and Marine Life Joint Industry Program. She was the chair of the National Research Council panel on ocean acoustics, education, and expertise through the National Academy of Sciences. She was also the recipient of an Office of Naval Research Young Investigator Program Award in 2011 and the Presidential Early Career Award in Science and Engineering in 2013. She is currently a fellow of the Acoustical Society of America. Her primary research interests are patterns and trends in ocean sea soundscapes, animal behavior and communication, and the impact of environmental change on marine life. She's authored or co-authored more than 60 peer-reviewed publications in a wide range of journals, including Science, Journal of Applied Acoustics, Progress in Oceanography, Journal of the Acoustical Society of America, Marine Mammal Science, and Ecological Informatics. Probably more impressive than that is she has received more than $25 million in grants and contracts from organizations like the Office of Naval Research, the National Oceanic and Atmospheric Administration, NOAA, and the National Aeronautics and Space Administration, NASA. Jen is currently working with the Shoals Marine Lab to install a cabled ocean observation system in the Gulf of Maine that will connect directly to shoals here on Appledore Island. It's my pleasure to introduce Dr. Jennifer Miksis-Olds, who will present her talk, Exploring Our Oceans Through Sound. Please join me in welcoming Jen.
Hi, can you guys hear me? We're good online. All right. Thank you so much for that kind introduction and for the invitation to be here. I did not know I was going to be the last Rock Talk, but I'm excited to close this seminar series off bringing this full circle back around to shoals, and I'm going to talk about that at the end. And so as Sarah said, if you have any questions or are confused or I use words you don't understand, I do this all the time and I forget that people don't speak sometimes the same language that I do. So just let me know and we'll stop and answer your question. All right, so we're going to talk about sound. You guys look really excited about this. But I'm super excited about sound because it's my whole world. So we're going to jump in and hopefully in the next 60 minutes or so, by the end of this, you'll be more excited about sound than you are right now. So if someone asks me, what do? I say I am a marine bioacoustician. What in the heck does that really mean? It's a very interdisciplinary field that I work in. So I have a background in classes with engineering for signal analysis, propagation modeling, hardware and software development. As you heard from my bio, I do a lot of marine biology. So I look at behavior, communication, and predator-prey relationships. I also have a degree in oceanography. So I look at biophysical interactions, I look at global climate change, coastal processes, and deep ocean dynamics. Statistics is there, not often very exciting, but you'd be surprised at how much progress and innovation is being done in statistics. If I could go back to school and do my career all over again, one of the things that I would have done differently is take more statistics classes. That's a weakness of mine, so I always partner and collaborate with people who have a strong background in statistics, and I still wish I could do it better. And if you work in the marine environment, either now or later, you're going to bump up against marine policy and regulation. So listen to all those birds out there talking about marine policy and regulation. I work with marine mammals, I work in the ocean, so everything I do has to have some sort of permitting in order for me to do it. If I put something on the bottom of the ocean out in the Gulf of Maine, I need a permit to do that. If I go and take a picture of marine mammals on a grant, I need a permit to do that. If I want to bring a cable up here onto Appledore Island, I need like 6 permits to do that. And so knowing your marine policy and regulation is really good to have in your back pocket as you think about what type of research you might be interested in. So the utility of underwater sound is awesome because it is capable of propagating over great distances underwater. Sound travels 5 times faster than sound in air. And if you're looking at low frequencies in the ocean, one sound made on one side of the ocean basin can travel all the way across to the other side. So all the way across the Atlantic or the Pacific if it's a low frequency sound. Global ocean sound can be used to monitor things like shipping and seismic exploration. People are like, well, why would we want to monitor shipping? Illegal fisheries. People are now Coast Guard and the Navy are starting to look at illegal fishing. They turn off their AIS systems or their automatic information systems that tells you where the boat is and what the boat is doing and who owns the boat. They could flip that off and then they're essentially non-existent on the water, but they can't turn off their motors, so we still know they're there. Seismic exploration is important. Those of you who like your cell phones and things made of carbon and plastic, having oil and natural gas is really important. And so knowing where that oil or natural gas is underneath the sea surface is done with acoustics. Climate change can be tracked through sound. Acoustic tomography looks at the average ocean of the temperature between point A and point B. And ocean acidification actually affects the way sound propagates in the ocean. So I just talked about all of the ways that humans can use ocean sound, but there's this whole other life of animals and marine life under the water. And hearing or sound is the dominant sensory modality for marine life. If you think about light, if you've ever been scuba diving, only the top few tens of meters have light. If you go down below that, it's essentially dark. So visual signals don't really help you in the deep, dark ocean. And if you look at light, it's not traveling horizontally very well. If you look at things like chemical signals, it takes a long time for a chemical signal to move from one place to another, whereas sound moves very quickly. So we as humans use sound to monitor the environment for military applications, environmental risk assessment and mitigation, We use it for things like earthquake or tsunami warnings to help people get out of the coastal areas if there's a tsunami off the coast, and to monitor for illegal nuclear explosions. There's a treaty organization called the Comprehensive Nuclear Test Ban Treaty Organization, where that is their mission, is to monitor both land and seas for nuclear explosions. And they do that with sound. So how do we use this sound? So the use of acoustic technology for understanding ocean dynamics. I don't only study life in the ocean, I study the ocean itself. And so active acoustics or sonar. You guys all familiar with sonar? Seeing anybody not? All right, sonar. Bats do use sonar, dolphins use sonar, and people use sonar. So we use sonar, for example, to do seabed mapping, to characterize the sediment on the bottom of the ocean, to analyze clutter, stuff that we don't really know what it is in the water column, and to image things like biological scatterers in the water column, things like zooplankton and fish. You guys have all seen, been on a boat or seen the fish finders, where you not only are seeing the depth of the water, but maybe speckles or stacks or layers of scatterers in the water. So that's your marine life. And then we've got passive acoustics. So that's just listening and recording the sound that is out there in the environment. From that passive acoustics, we can look at things like sea ice dynamics, wind and precipitation rates, marine animal detections, environmental sound levels, and detections of human activity like shipping. So I'm going to show some of these things on the bottom left-hand side. They're called spectrograms. So let's see if I get a cursor here, maybe. Oh, there we go. Can you see my cursor up there? No, you cannot. Can I? Oh. Okay, well, the x-axis on here is time. So we're showing about 4.5 seconds of time, and the y-axis is frequency, going from 0 to 6 kilohertz. We hear, as humans, we can hear from about, I don't know, 200, 300 hertz all the way up to, if you're young and haven't ruined your hearing yet, about 16 to 20 kilohertz. If you're old like me and have listened to loud music, I can hear maybe up to 10 kilohertz. So it depends. And I'm going to play you a sound, and it's got 3 different animal groups vocalizing. We've got a ribbon seal sweep, we've got bearded seals trilling, and we've got walrus knocks. So this is a way, these spectrograms are a way for us to see what we're hearing. And I'll play that sound for you. Maybe. Did you guys hear all 3 of those different animal groups? Can you turn it up a little louder? Okay, here we go. There we go. So I could pick those out very easily, but the students that come in my lab to help analyze data, we go through a training process. After you listen to a few thousand of these, you kind of get the idea, and you can pick them out actually without even having to see them. There we go. So over my career, I've had really 3 different research foci. Everything's under sort of one umbrella of acoustics, but I use acoustics to understand how the oceans are changing, like climate change and soundscapes in the ocean. How are animals impacted by environmental change? So I look at response to anthropogenic effects or human factors, such as rising ocean sound levels from ships, or pile driving, or seismic air guns for energy exploration. And I look at biophysical interactions. I also look at the behavior. I use acoustics to look at the behavior of animals in their natural environment. So it's really hard to study marine life biology and communication, because most of the time you can't see them, they're underwater. So acoustics allows you to do that. So we're able to look at communication, perception, and cognition, and things like predator-prey relationships. So I'm going to talk about 2 projects today. One is called ADION, the Atlantic Deepwater Ecosystem Observatory Network. This was a 5-year project that happened on the outer continental shelf between Virginia and Florida. It is over now, but all of the instrumentation that I'm going to talk about that we developed during the project has now been moved up here to the Gulf of Maine and ties into the next project related to the cabled array that we're going to have installed hopefully this fall. That's going to come on here to Appledore Island. So ADION was a project with the challenge of monitoring both the mid and South Atlantic outer continental shelf from about 100 to 1,000 meters. We needed to make multi-year, year-round measurements. And so we did this with an integrated system of multiple platforms. We had fixed platforms called alto landers, and I'll show you what those are, mobile platforms, which was a sailboat, and we had remote sensing that we used from data from satellites. So we were able to use and integrate measurements related to the soundscape, biology, physical oceanography, water quality, air quality, and also human factors. And one of the requirements for getting this project was to come up with an overall system that was both relocatable to other regions and generated data that was comparable with other projects in other places at other times. So it was an extensive project. And in order to do that, I listed all the names of the co-PIs that we had on this project. This is not something I did alone. This is something that we do in research teams because all of these people brought expertise to the project that I did not have myself. And so teamwork is really important when it comes to oceanography, especially offshore oceanography. So here was our network design. We had sampling and data platforms. You can see the sailboat, that's our mobile system. We had subsurface landers there on the bottom. We call them alto landers, acoustic long-term observation landers. We use space-based remote sensing. That's the satellite up there where we looked at chlorophyll data, sea surface temperature, primary productivity, precipitation, mixed layer depth, wind and currents. That sailboat had a towed array, so a towed line of hydrophones to listen to the ocean, to record ocean sound. We had vessel-based sampling. That's the boat on the right, where we did net tows and acoustic surveys, and I'm going to talk and show you some of that data. And then we had ancillary ocean observing systems such as AIS, which are the automatic identification systems on boats. So we brought all that together in a network to integrate all that information to ask ecological questions. This is an alto lander, long-term autonomous observatory lander. These were designed specifically for this project. So we kicked those off the back of a boat with a crane, and we just let it drop to the ocean by itself. And it was weighted appropriately, so it landed right side up. And so I'm going to start on the top left here, that CTDO, that is conductivity, temperature, depth, and oxygen sensor. We have a satellite beacon on there. So if it surfaces when it's not supposed to, or we release it and it surfaces and we can't find it, it lets us find our data and get our data back. The hydrophones, there's 4 of them underneath those little yellow flow caps. We have 2 acoustic releases on there, which we send a message to when we want the lander to pop up, and it drops the anchor and floats up to the surface. We have a fish tag receiver, so anything that is tagged like a cod or a great white shark or hammerheads that people have tagged for different types of projects, if they swim by our lander, we get a recording of that. And then those black boxes on the front are our sonar systems, our echosounder systems. And I'm going to talk more about that when we get to the data analysis part. Phase 2 of our project was data acquisition. We went on cruises for 4 to 6 weeks each year. We did lander deployments. We had the sailboat doing towed array missions. We had the boat do this lawnmower pattern to look at all of the fish and zooplankton backscatter from our echosounders. We did CTD, so temperature and salinity netcasts, and we had active marine mammal observations. And we did this for our 7 different sites. This is just one of the cruise trips that we had where we left Woods Hole, Massachusetts, traveled down, serviced all 7 landers at all 7 sites, did our nighttime sampling, and then went back. Takes about 4 to 6 weeks, depending on how fast you go and how much bad weather you hit on the way. So here's some of the data that we recorded. On the left-hand side is an example echogram from the ship's echosounder on the bottom of the ocean, looking at internal waves. So those tiny little waves that you see up in blue there, those are internal waves, waves under the water with temperature salinity gradients. The red squiggly line at the bottom is the bottom. The ship's on the surface and it's imaging all the way down to the bottom, and that shows you your bathymetry. And at night, when all of the zooplankton and small fish that were at the bottom during the day migrate up to the surface, we did this fine scale acoustic survey with the ship's echosounders, where we did this square pattern. We went back and forth with the lander being in the middle, which is that red dot. And you have to remember, the lander had echosounders on the bottom looking up. So we were using instruments looking down and instruments looking up in order to get the entire water column. At night, we also did our net tows. We did bongo net tows and IKMT, which is, Isaac Kidd's midwater trawls. The IKMT net is on the right-hand side, middle there. It's big and you need a crane to deploy it. But the small ones for the small zooplankton on the left-hand side are easily just put over by hand or by a winch. And you can see some of the zooplankton and nekton collected while we were out there. These were all taken through microscopes. So really different types of organisms that would come up in our net tows. I'm not going to talk about the data processing. I'm going to talk about the data results, but I do want you to know that if you look here, you've got data type across the top. We had our omnidirectional acoustics. We had towed acoustics. We had fish tag loggers, chlorophyll, marine mammal observations, net tows. We had all this data coming in. And then you see the data products on the left-hand side. So things that we were able to discern or study, different aspects or parameters about the ocean and the dynamics in these areas. And we were able to look at those and compare them in between sites. And I'm going to show you some of the data that has come out of that. So let's start with sound levels. We had 7 sites. They're on the right-hand side, Virginia on the north, all the way through to the Blake Plateau off of Florida in the south. Frequency is on the x-axis, and amplitude, or how loud the sound is on the y-axis. So take a minute, all 7 sites, these are averaged for a year. Which site would you say is the loudest? All you high schoolers, SAT question. This is something that you'd get for, understanding graphs. College students, you're going to be answering this in your labs. Who wants to venture a guess? All right, go ahead. The blue dotted line, that's the Virginia. And I would agree with you for frequencies below 1,000 kilohertz. So frequency matters. If you look on the right side of the graph, sometimes that purpley line comes up and is loudest. That's the CHB Charleston bump. Which one's the quietest? Raise your hand so I can. Which one's the quietest? Go ahead. Red dotted line, Hatteras. Yes, I would agree with you for frequencies above about 200 hertz. But that green one at Savannah, SAV, is definitely the quietest at the lowest frequencies. So here, what I want you to sort of take home message from this is that sound is interesting in that depending on the frequency, you may see different patterns and trends. And that's because different sources make signals or produce sound at different frequencies. So let me ask you a trivia question. Blue whales, high frequency or low frequency sources? Low frequency sources, good. Echolocation of a harbor porpoise, high frequency or low frequency? Okay. How about a croaker, a fish? Oh, not so confident about that one. Yeah, low frequency, I would say below 1,000 hertz. How about a large tanker ship? Low. How about a jet ski? Higher, mid-frequency. So now you're starting to think, bigger the sources, the lower frequency they are, the smaller the sources, the higher frequency they are. All right. This is a lot of data. This is from one of our papers that was in, I think, Marine Mammal Science now. So it has marine mammal group on the left-hand side, and it has the stations on the right-hand side. Again, arranged north to south, Virginia in the north, all the way to the Blake Plateau off Florida in the south. I'm going to start asking you questions because there's so much in here that we have learned from this. These are monthly presence absence. So if you see a dot, it means that that animal was there that month. And this goes from 2018 to 2020. So we've got like 2 years of data here. Which animal group is most present through these 2 years at all sites? Dolphins. All right. Which animal groups or animals do you see clear seasonal patterns? Minke whales and fin whales. Very nice. Okay, good. All right. Do you guys know what the term site fidelity means? So site fidelity is when an animal or an animal group is constantly present, but only at one location compared to multiple locations. So dolphins were found everywhere. They don't have high site fidelity. They were found at all 7 of our sites. Which group of animals showed very strong site fidelity? Blue whale, they were found at 1, 2, 3, 4 sites. So I would not say blue whale. Another one? What was that? Couldn't hear. Shout it out or raise your hand. The one above the sperm whale? Kogia species. They're at 2 or 3 sites. Which species there are only at a single site? Cuvier's beaked whales. Yes, what else? Bladesville's beaked whales. Which one? Right whales, we only saw them at 1. Yes. So you're starting to see now from this data where we can start to look at site fidelity and where animals are naturally found. Before this study, we did not know that minke whales were using the southern outer continental shelf as a mating ground, but this clearly shows seasonal migrational patterns to those areas. And now we know that they occur there. We see for beaked whales, so those are hard whales to see at the surface. They're very cryptic, but depending on the different species of beaked whales, they're only at sites. So it's really interesting and new information to know that beaked whales exhibited fidelity on the outer continental shelf. So I could stay here. We could talk about this for a while, but I'm not going to do that. We're going to move on to source directionality. So last time I showed you something that looked like this with time on the x-axis and frequency on the y-axis. I called it a what? Spectrogram. Absolutely. This is a directogram. So now I'm going to change it up. Instead of the colors indicating amplitude, they indicate direction from which the sound came from. So red is coming from the north, turquoise is coming from the south, yellow from the east, and bluish from the west. So if I had to put a spectrogram here, you would never know that there were 1, 2, 3, 4, at least 5 minke whales vocalizing in this 4-minute period. You wouldn't know that it was 1 whale or 5 whales. But with the directionality from 4 hydrophones, we're able now to know how many animals are vocalizing at the same time. which allows us to get one step closer to estimating how many animals are out there at any time. I'm going to just stop here and take a step back. When I was a graduate student, I wanted to know how many sperm whales were out there. I wanted to do my dissertation on looking at sperm whale abundance in reaction to anthropogenic sound. And my committee said, there's no way you will ever be able to know how many marine mammals are vocalizing, because you can't distinguish between one really chatty sperm whale and 5 less chatty sperm whales. 20 years later, with advances in ecological statistics, we are now able to do density estimation from passive acoustic monitoring. So something that I was told by my committee when I was a graduate student 20 years later is now possible. They told me it was impossible and I couldn't do it. And I didn't do my thesis on that. 20 years later, it is possible. So things will change in your generation, and it's kind of exciting to be part of that information change. So now I'm going to move to active acoustics. So these are all echograms. Here we've got a day, 24 hours on the x-axis, and on the y-axis is the depth of the water column, the surface at the top, the sea bottom on the bottom. And we've got 4 different frequencies. What do you notice about this same parcel of water being imaged over 24 hours with the 4 frequencies? What do you notice here? I'm asking you to think. You couldn't even type this into an AI question and get the right answer. What do you notice? See, I got all night. I can stay here. So you're looking at the same image of water over 24 hours at 4 different frequencies of sonar. It would be like having a fish tank and shining 4 different frequencies of light through it. All right, hand over here. Yes, as the frequency goes up, we kind of lose signal on the graph. That's good. What else? Go ahead. The y-axis is time. No, that's the x-axis. Y-axis is the depth of the water column from the surface to the bottom. It's like a fish tank that you're looking in right now. So one of the things that's really clear, if you look at the big blob in 38 kilohertz, it pretty much disappears in 125 kilohertz. You see different features with different frequencies. So the great thing about having multiple frequencies is that you can look at the ratio of the echoes from one frequency to the other, and you can start to attribute those differences to different animal groups. So for our data, we were able to, with our 4 frequencies, discriminate small and my cursor doesn't work. Small animals, less than 20 millimeters. Medium animals, 10 to 40 millimeters. We have these large resonant scatterers. Those are things with like swim bladders or air pockets. We've got large resonant scatterers, which are, again, swim bladdered fish. And then we've got small resonant scatterers like siphonophores. So we're able to start to look at our active acoustics and assign some sort of animal community structure to it. So if you look at a year of data here from one place, you can see in the beginning of the season or beginning of the time series here in December, the orange is dominating. So that orange is the non-resident scatterers. So those are things like copepods and euphalcids. As you get towards the summer months and into the fall, you start to see the yellow line dominate, and those are large resident scatterers, so fish. So what do you think's happening here? What's happening to all of our copepod and euphalcids that are sort of coming into our community first? What's happening to them? They're getting eaten by these fish in yellow. So yeah, so you're starting to be able to look at community structure changes by only using sound. So I showed you a lot. I can't show you everything for this project, but if you're interested in learning more about this, all of this data is free and available to the public. We have a cool website with visualizations where it shows all 7 landers, and you can click on a lander and it shows all the marine mammals that are located there. You can click on different buttons on the left there and bring up wind and chlorophyll and sea surface height and sound. And one of those things I like always to show the hurricanes. So we watch the hurricanes and the sound change at each of the different locations. This was Hurricane Florence in 2018 passing over the lander. So you can look at that on your own. You can look at sites and you can play the noise or the sound, I should say. So the top is about a week's worth of data, the middle about a day, and the bottom about, I don't know, 10 minutes worth of data. And if you go online, you can actually hit the play arrow and you can listen to all of these things. So any idea what those large spikes are in the middle? What are those loud, bright sounds there in the middle? Any idea? Boats, ships, yes. What about those low frequency speckles on the very bottom? What are those? What was that? Not wind. Not fish. Getting closer, you got biology. So whales, those are fin whales, which are the rats of the ocean. You always find fin whales places. So yeah. And then you can start to look at these detections. So right now I've got up here, it says sea surface temperature. Those are the colored ribbons on the top and the bottom for, what is this, for a year. This is from 17 to 18. All of the marks on here are dolphin clicks. So you can see where they occur throughout the year. The different colors are the different years. The gray middle is nighttime, and the light colored periods are daytime. What's one take-home message you can get just by looking at all of the dolphin clicks over this year? One take-home message that you can get from this data. They are talkative. Yes, all year. That's good. What else? Oh, go ahead. Oh, yep. The wave in the middle, that's the nighttime hours. What was that? Those are all detections of dolphin clicks. The blue are detections of dolphin clicks in 2018. The different years. They're more active during the night. Exactly. Look how much more active they are in the night than they are during the early morning hours at the bottom. So yeah, so now you're starting. What about, could you look at the relationship between dolphin clicks and sea surface temperature, which are the colored bars? Where do you see the least amount of clicks in here? Bottom, but that's just daylight hours. But we're going from left to right is a year of data about. Right early. So yeah, there's less on the right when it's warm. You see more in the middle when it's cold. So you can start to play with these on the website. You can look at fin whales, and you can look at dolphin whistles, and you can look at minke whales, and you can look at you can change the color bars from sea surface temperature to chlorophyll, for example. And you can start to play with all this online. One of the things that we did during this cruise is we had 2 artists come out with us. This is artist Wendy Klepperer. She's a sculptor. For those of you on campus that are going to be going to UNH and being freshmen, you're going to see large sculptures on campus. Those were made by Wendy Klepperer. She also does a lot of watercolors, and she came to see with us. And we had a nice art debut on the bottom right-hand side on the last day of being at sea. You can see her here. We had the UNH Museum of Art actually had a huge exhibit of all of her stuff while she was at sea. So we loved having her out there. This is another set of artwork from Lindsay Olson. She came out with us. On the left is a tapestry of her rendition of passive acoustics, and on the right is active acoustics. So you can kind of see the different zooplankton and fish layers there in the active. She actually made her own beads when they weren't the color and she couldn't buy them. And later, if you want to come up and look at this, all of the ocean acidification and wave equations, all the math that goes into acoustics, she's used and embroidered within and around all of the art. So it was a really nice combination of art and science. And these will be hanging for those going to UNH in Morse Hall on the 3rd floor. I just purchased those and they're being donated to Morse. So you can see them anytime. And I also learned, this is something that I learned, that oceanography is a team sport. When we went out there for our cruises, we found all sorts of weird things. We found a disabled autonomous sailboat. We found a capsized rowboat, which is always tough because you never know what you're going to find when you go and turn it over. We found a glider that lost its way. We picked up a CTD off the bottom of the ocean because the ship lost it. So we did a lot of finding of things. But karma is often returned. We lost a lot of things. So this lander, for some reason, we don't know why, surfaced and started to head towards Cuba. And we were able to find another vessel that was down there and was able to go get it for us before it crossed the international line. We had a lander that got stuck in a sand wave. So we had to collaborate with ROV Jason here out of Woods Hole to go down and clip that little line on one of ours and dig the lander out of the sand wave. And again, it's a team sport. I don't do this alone. These are all the people on the different cruises that we went. So now I'm going to switch gears real fast and talk about the cabled array. So that lander that I talked about, that we had 7 of off the East Coast, we now looked at the Gulf of Maine, and this is about 20 years of data of people looking for right whale vocalizations in the Gulf of Maine. You see a lot of detections close to the shore. You see a lot of detections on Browns Bank and in the Canadian waters, but you see a huge hole in the middle where we just don't have any data. So what we did is we took our landers and we filled in some of those holes. So there are now 5 landers in the Gulf of Maine, all within acoustic connection with each other for low frequency. So that image on the right-hand side shows a propagation of acoustics of 100 hertz. And you can see that a signal on right near one lander can easily be picked up on the other lander. So we're covering the whole pretty much Gulf of Maine with our 5 landers to listen for low frequency sound. So those are the landers that are out there. We also had funding to put in a cabled array. And so this is where it's going to be. So this should look familiar to you. This is Appledore Island here. We're going to have about a 3 to 5 mile cable extending from Appledore Island in the ECB building here to this equipment platform. It looks like a lander right there on the right-hand side. It's going to be approximately 15 kilometers from shore or so. And that's going in October. So when that goes live, all of the data coming in from that lander will be coming in real time and available on the internet. So you guys can watch what's going on in the offshore waters east of Appledore Island when you go back to your own schools. So here's the concept. It's got an underwater sensor platform, it's got a cable, and it's got the island station and all the racks and computers. This is what that's going to look like. This is the underwater sensor platform. You can see all sorts of sensors on it. Instead of me trying to point these out to you, I'm going to point them out on the schematic. So you can see we're going to have 2 cameras on it. This is one that was deployed in Boundary Pass in Canada. Got some really cool footage of a foraging octopus, and we hope that we get some footage that's just as cool. We've got the fish tag receivers I talked about, the echosounders. We've got temperature, salinity, dissolved oxygen, chlorophyll. We've got a current profiler. That's the ADCP, acoustic Doppler current profiler. We've got our hydrophones, and we've got 2 cameras and a light. So all that is going to be streaming data back to land through Shoals Marine Lab. One of the cool things that we're seeing in the Gulf of Maine with our lander data, these are all sei whale vocalizations. And you can see that there's this, the direction is changing gradually over time here. So this is a directogram. And it's actually in this box here, you can see a sei whale transit through the area past this lander. The cool thing that we can do, again, with this directional data, you see the bearings on the bottom. That tells you which direction it's coming from. That whale came and went. You see the detections per day. That's at the top. And in the middle, you see this absolute minimum number of animals per day. So just from the directional information, we can get an idea of the number of animals per day from each of these landers. You can start to look at migration patterns. Which way are they coming from? Which way are they leaving? You can look and see if they're uniformly distributed across the season. Gray is fin whales. I told you they're rats of the sea. They're coming from everywhere. They're everywhere. What animals do you see kind of disappear or only are there very briefly? The ones in green, those are humpback whales. Sei whales are blue. You only see those sort of there in the summer when all the fin whales are kind of gone. So now you're starting to see niche partitioning and different feeding behaviors of the different types of large whales. Back to the active acoustics. On the bottom left-hand side is a year of backscatter data. So you can look at seasonal blooms of different sizes of animal scatterers and fish. You can look at diel vertical migration, which is shown up on the right-hand side, if you're interested in daily patterns and trends. So let me just say that putting a cabled array in is not easy. There are a lot of challenges in order to get data transfer from the actual sensor platform to the ECB building here on the island to the mainland. As you are all aware, Appledore Island, Shoals Marine Lab, is a green island. So we need to have a lot of power limits. And how do you integrate something that's power hungry into a green grid? So we had to buy a lot more battery banks. And you'll actually see out there, I just saw it today from my eyes for the first time, the new solar panel banks. I was like, yay, look at that. And then we had supply chain challenges. Still from COVID, we're still waiting on pieces that we're hoping arrive before October before we have to put this in the water. And I just want to finish with permitting and approvals. This is a $6-7 million project, and nothing is going to happen unless you have permits. We needed 1, 2, 3, 4, 5 permits on the land side. And then we had to have permits that are in compliance with national laws on 1, 2, 3, 4, 5, 6 different laws on the water side. So take a guess on how long it takes to get all these permits. What? A couple of years. It's taken about 2 years. And we still don't, we have all the land ones because the land work has started, but we do not have all the water ones yet. And I am crossing my fingers that they come in before October when we're supposed to have the cable laying ship coming and putting that cable in. So yes. I just want to say again, I keep pounding this out that oceanography is a team sport. We do it in teams, and not only the people doing the work, but the people funding the work. So this work was funded by NOAA, by BOEM, Bureau of Ocean Energy Management, and the Navy, Office of Naval Research. The National Oceanographic Partnership Program helped make that happen. And then we have all of our collaborators there for the cabled array and ADION project on the bottom. So I'm going to leave that there and answer any questions. Ooh, yay, I was on time. That never happens.
So we do have a microphone for anybody who has questions in the audience here in the room. We'll also be checking to see if anybody we already have a question online.
All right.
Jackie Webb is asking, the project in the Gulf of Maine has just started, but based on changes in species composition and abundance in the Gulf of Maine over the past several decades, what changes in soundscape of biological origin are you expecting?
We're now 3 years into the Gulf of Maine project. We have actually got 3 years of two plus years of data back. So we're still processing that, but what we are expecting, one of the things we're expecting is to see changes in migration, arrivals and departures with animals, because global climate change is changing some of the environmental parameters that govern their prey. And so as those prey dynamics change, we expect to see the marine mammal dynamics change, which will change the soundscapes related to marine mammals. With global climate change, we're starting to see the opening of the Northwest Passage between the Pacific and the Atlantic Ocean, which means when ships can come through, that Portland, Maine is going to be our first deep water port in the US for ships going through that Northwest Passage. So we would expect to see shipping signatures and acoustics in the soundscape change related to that as that starts to happen. So those are two examples of things that we're expecting to see over the next probably decade as things change in the Gulf of Maine.
Anyone in the room? I'm going to be bringing the microphone around so that the people on Zoom can also hear.
Oh, there's two more. So the people on Zoom can hear the microphone? Excellent.
Hi, Jen. My name is Jenna. I'm on staff here. Thank you for your talk. This was super interesting. With the first project, and I guess kind of leading into your second one, what sort of kind of end results? You showed us a lot of the data, but did you have any like policy recommendations that you were doing or papers that came? Like what sort of was the big outcome of it?
Absolutely. So we had multiple papers that came out. And I just pulled, for example, images from some of those papers. And I talked about, let's go up to, so we have one paper out that looked at changes related to COVID, for example. And we found very little change related to COVID in the offshore waters compared to other studies that had shown huge decreases of sound in coastal waters. And so that long distance shipping was still happening and trying to meet supplies. So we have a paper on COVID. This paper was in Marine Mammal Science, really looked at and highlighted the new knowledge of niche or site fidelity for different animals, and the fact that we were able to show that minke whales, for example, were using specific areas of the outer continental shelf for mating and breeding. So that came from there. One of the papers that we had that came out of the active acoustics looked at comparing the data recorded from the vessel's echosounders as it mowed the lawn. Which one? There. Compared to the stationary data looking up. And by combining them both in the movement of the ship, we were able to define how far from a lander you could assume that those characteristics of a water column or animals would be representative. And that showed to be about 1 to 2 kilometers, which was the same for all 7 sites. We're doing the same thing now up in the Gulf of Maine to see if a different region has different distances of extrapolation and you needed to have a mobile and a stationary one to be able to do that. And so that's some of the data, one of the papers that came out of the active acoustics. We had a paper come out of the soundscape modeling work from Adion. Yeah, sort of like this. So this is a soundscape model, and we were able to progress soundscape modeling by including ships and wind and be able to hindcast and forecast. And so with this data, we were able to validate the models and help refine them a little bit more moving forward. So we have different people working on different aspects of these different projects. So multiple papers came out of this. And there are papers still coming out of this. We used, I had a grad student here, a master's student, who looked at 2 of the Adion sites, which were deep water coral sites, and compared the soundscape from there to the deep water sand habitats. And then we're able to compare those to shallow water corals. And so we're able to look at some of the differences in the defining characteristics of shallow versus deep water corals and some of the ecosystem dynamics that make them similar or different. Sorry, long answer to your very simple question.
I think somebody else had a question over here.
Yep.
Lot of stuff to think about there. Thank you.
Yep.
So this is maybe a little out in left field, but do we know if microbes care about sound?
That's a good question. All marine animals have been shown to have sensory organs to detect sound, even if they don't make sound themselves. Microbes are not technically animals. I don't know the answer to that question. I know that large masses of microbes do scatter sound. So, yeah, you don't care about that. You care if they could set. Did they care? You know, that's a great question. And to tell you where the field is, we are still trying to, as a community, not me personally, wrestle with the question of whether zooplankton care about sound. And so we haven't even been able to answer that question well. There have been studies that show that zooplankton react to sound in to like seismic airgun arrays. That was shown by colleagues in Australia, but there have been other studies that show that there was no impact to sound. So it depends maybe on the region, on the species, and so forth. Fish, there's a lot of work that's shown that some fish, especially hearing specialists that can hear a broad range of sounds, do react to sound, whereas the same sound in the same study for fish generalists that don't have a connection between their ears and their swim bladder don't react to that sound. So it's very context specific, but we haven't really, as a community, been able to address that question of microbes because we haven't quite even been able to address it with zooplankton yet. I saw another hand over here.
How does like being deaf or hard of hearing affect marine animals, especially like whales and dolphins and animals that use echolocation?
That's a
Things like that.
great question. And people have looked at that. Animals that have temporary threshold shifts in relation to loud sound, there's temporary threshold shifts where your hearing recovers. If you think about it as a human, if you go to a loud concert and you don't hear very well when you get out, that's a temporary hearing threshold. You'll wake up the next morning and it'll be back to normal. Permanent threshold shifts mean you lose hearing. And studies have shown that temporary and permanent threshold shifts impact an animal's ability to evade predators, forage for fish, navigate, and so forth. That's a great question. Hearing is remember I said hearing is the dominant sensory modality of underwater life. It's like our vision on land.
Do we have any other questions in the room?
This might be a little bit silly,
No
but
silly questions.
I was really interested when you said that people have to listen to sounds for a really long time before they can identify them. What do you do if there's a sound that like no one knows what
Oh,
it
I
is?
can't tell you how many sounds there are out there that people don't know what they are. And here I'm gonna, you know, swallow my pride. And so this sound here, this one that says ribbon sweep, it goes. When I first heard that, comes from the Bering Sea. I'm like, it sounds like an arcade game. I was like, what the hell is this thing? And so I didn't know what it was, but other people did. There are listservs, groups where you can post a sound and say, what is this? And someone will, if you've heard it, come back and tell you. There are groups of people that we know there are some sounds that are baleen whales, but we don't know which one. And so there are groups of us that have tried to get together. I'm on a paper that's in review right now, looking at some new blue whale calls. There are always sounds out there that we do not know what they are. But a good resource for learning sounds and what they may be, there's a website called Discovery of Sound in the Sea, DOSITS, Discovery of Sound in the Sea.org. And that has an audio gallery of human sounds, marine mammal sounds, fish sounds, weather sounds. Lightning's really loud, for example. So that's a great place if you want to start to learn about sounds, that they have a sort of a dictionary there.
So that's all the time we have for questions, but I'm going to
Oh.
give Jen one chance to say something else because we do have a large number of people at UNH. We were speaking this evening about, I'm going to get, so we don't have feedback. We were speaking this evening about an event that happens every spring at the UNH campus.
Yes. So we have at UNH an acoustic symposium, which is one day in the spring, every spring, where students and faculty from the region and outside the region come to present their acoustic data. It doesn't cost anything. And the neat thing is that the next day we have an acoustic career symposium. So people from academia, from federal labs and government, from Department of Defense, from local and even international industry come and share how acoustics is used in their industry. And they actually recruit students right from that acoustics career workshop. We have had students get jobs before they left. It was amazing. And so especially for undergraduates in ocean engineering, or I mean, the different groups all look for different things. So it's an event, it's a 2-day event. One is technical research day and one is career workshop day. So that happens in the spring. So watch our Center for Acoustics Research and Education website if you're interested in things like that.
And with that, I would like to give one more round of applause for Dr. Miksis-Olds.
I have to ask one question now. Sound is my life. I'm very excited about it, as you can probably tell. Are you guys a little bit more excited about sound than you were when you started? Awesome. Then I did my job.
And so hopefully you will see sound differently is kind of what I was going to say. So thanks again. And I think everybody knows what they're doing next. And this is the end of our Rock Talk series for 2024. Thank you all for being a great audience.